Okay, so we are just about ready to begin. Let me try to make sure that I've given enough context about what it is that we're doing, and then we'll go ahead and try it. So the first thing is we're actually following, there are some protocols online. There's something that Nanopore has released called NOMIS, Nanopore Only Microbial Isolate Sequencing, something like that. I'm following something slightly different. I'm inspired by it. But in my case, I'm going to be using a ligation protocol. So in other words, we're going to be sharing the DNA and then using a ligation method to attach adapters. So that's going to make for a longer protocol. I really like the ligation protocol. Sometimes the rapid protocol, which is shorter, yields different results. It's really, you have to try it and see on a case-by-case basis to come up with what works best for you. But because I haven't done a ligation protocol for this video series, I wanted to specifically do that. In my case, I've got 3 soil isolates, and these are isolated by students following a different microbiology procedure. And now I have the cells, and we're going to go ahead and use one of the Monarch kits. So this is a pretty general purpose kit. This is a Monarch high molecular weight DNA extraction kit for tissue. And on the protocol on the website, we'll have appropriate links to things. I've got my protocol printed out, so I think I'm ready to go. Because I am working with theoretically unknown bacteria, you need to consider what is the appropriate protocol and PPE in your situation. I don't think it's quite that dangerous at all, but I am wearing gloves, of course, and I am also wearing a little eyewear. I mean, the real danger, since I'm working with a number of cells, you know, might be if I got something in my eye, or if I had an open cut, or something was aerosolized. So you might want to be a bit careful working with this type of sample. You can consult with, if you don't already have the microbiology experience. And then I would say in an educational setting, one of the things you could do is there are certain parts of the isolation protocol of going from an unknown soil sample to a cultured and high concentration of an unknown bacteria. There's steps that you could elect for students to do, and then other steps down to the point at which you neutralize the bacteria, which we're going to do. We're going to lyse them in a second. That would allow you to make sure that you're not working with any live cultures if you want to be extra cautious. I'll say that I do happen to know that these bacteria are gram-negative. And so there are 2 options for bacteria, gram-negative, gram-positive, according to the protocol that we're using. And so I'm going to stick with the version, which happens to be, thankfully, a bit simpler in terms of lysing for the gram-negative. So the first thing I'm going to do, I've got 3 isolates, number 4, 19, and 20. And I'm going to take some of those cells and go ahead and pellet them down. The protocol gives you an idea of how many cells you want, so you can go ahead and get an estimate of that with spectroscopy to get a sense of how many cells that you have in your liquid culture. I would say if you're not sure, then usually a lot of times less is more. Whoops, didn't need to do that. A lot of times less is more, so don't be afraid to go with a smaller sample if that's what you have and you're not quite sure exactly what you've got. These are quite colorful bacteria, so that is of interest. And again, the whole part of the isolation and characterization and gram staining, each of those things is potentially something that's interesting to do with a student. These have been frozen for a while, and this one is quite interesting because it's quite viscous. So I'm going to do my best to get about 1 ml out of that. I happen to be using, I don't think it's particularly required, filter tips, more because I want to protect. I'm going to use this set of pipettes for everything, and a lot of times I just want to make sure that I am keeping things I'm being really good at separating my materials. So this is a quite viscous sample. I'll pay attention as we do various things to try to make sure we're accounting for that. Okay, so I'm going to go ahead and complete just transferring these over. The colors, are important. Actually, in the case of what we were trying to do with the students, we were interested in seeing if we could isolate soil bacteria, potentially ones that might produce antibiotics. So in theory, right, there is an interest in the applications of microbiology, the fact that you could find bacteria that might produce a novel antibiotic. That's how a lot of the first antibiotics mass-produced were found. Okay, and sample 20. I'm only doing these 3 samples, these 3 isolates. The barcoding kit that we're using would allow you to do up to 24. Now, some of the things to think about in choosing how many you're going to process kind of goes around the idea of the more DNA that you're going to be sequencing in the barcoding kit, you're going to be dividing your total number of reads, your total output from that kit by 24 if you were to do all of the possible samples. So you'd have less coverage if you're trying to do a full genome assembly. I'm using more, so hopefully that means I'm using fewer samples, so hopefully it means I get more coverage or better chance at complete genome assembly for these samples. Okay, so I've just transferred them. That wasn't very exciting, but what I do need to do is spin these down and pellet them. I'm going to resuspend them in cold PBS. So I will go ahead and do that. Let's see if I can balance this a little bit better. There we go. I'll mention, with a view to people doing this in the classroom, things like the cold PBS. If you don't have that lying around, it's different if you're working in a molecular biology lab. This particular kit calls for a couple things, absolute ethanol, PBS, but not too many more things. A lot of the stuff is included. In fact, actually, it's worth mentioning that the RNAse A and the proteinase K that comes with your kit, it's going to be a good idea to, as soon as your kit comes, they are fairly shelf stable, but they need to be stored at -20. So make sure you open, if you order one of these Monarch kits, make sure you open it as soon as you get it and pay attention to the storage instructions. It's not uncommon that there are some items in that kit that need to be stored at a different temperature. In our case, because we're using these gram-negative, all this really is going to call for is lysozyme as the primary agent for doing the lysis, so that's good. You need to, again, check the kit and also consider at this lysis step, you may not know what bacteria you are culturing. You didn't do the gram staining until, after you cultured things and see what you got. So be prepared that there are other ways to lyse the bacteria, and depending on what you want, certainly if you apply protocol and there are bacteria in your sample that aren't lysing with that protocol, you're not really going to get what you're looking for. So be aware of the sampling biases that could happen at this particular step. Okay, so I have spun those down, and now what I'm going to do is I'm going to pour off that supernatant. Let me be careful because one of those was viscous, but I'm going to hopefully spin it down. Now this one actually is a very good example. Okay, these pelleted down really, really nicely. You could see a gigantic pellet in there. This one, because of the viscousness, did not. So what I am going to do is I'm going to spin that down just a little bit more. I could consider at a higher speed, or consider what I'm going to do, just because of the fact that it did not spin down as well, which is not surprising. It's only 1 minute at maximum speed. I do have centrifuges that can go for longer, so we'll see if that helps. to do it for another 2 minutes or so. I'll come back to you when I'm done. Even if it doesn't, I'll take what I have and we'll think about that as a low input. We should see it reflected in the throughput that we get later on, but we'll just go with it. But I'll take note of what I saw. So let's see what happens after 2 minutes. Okay, so I spun that down a little bit longer, but it really is just a very viscous sample. So I'm gonna not worry about that. I'm gonna go ahead and proceed. But this one, the other one's pelleted beautifully. So we'll proceed, and I'll think about adjusting some levels of things, but we'll go with what that is. So I've done step 1, and now in step 2, I'm following the protocol and the portion of it that is for working with gram-negative. So I'm going to go start by resuspending those that are in need of resuspension in some cold PBS. Let me actually grab the right pipette. That make a gigantic difference. We're going to use 300 microliters. This is the high input. And by and large, I'm trying to have some semblance of sterile technique. Cross-contamination is more of an issue than actually worrying about contamination with whatever environmental bacteria are here, because this is a pretty overwhelming sample. That what we end up with is going to be the bacteria that we are hopefully looking to work with. And I will not be worrying about doing this for the one that I can't. Now, the next step is going to be to add some lysozyme. And again, this is something you can make. Some people buy lyophilized. But if you're not using it all the time, you may want to go ahead and make sure you buy lysozyme that, in fact, I think lysozyme might be one of the things you have to check whether it's included in the kit or not. But lysozyme, you can buy lyophilized and dried and make it, or you can already make one that's approximately whatever concentration they want. Of course, they go ahead and they sell that for you. So you can do that, and I've got lysozyme here. And what I am going to do is add that, and it requests 10 microliters. There's all sorts of different types of lysozymes, and you can make some adjustments. I'm actually going to add a little bit more than 10. I'm going to add double, and the reason why I'm adding 20 is because in the next step, the way that they envision for you to do the lysis on a water bath or in a heat block with agitation, and I don't have one of those with me, so instead I'm going to be relying upon my fingers to do the agitation. So in my own head, I'm imagining that adding a bit more lysozyme will give me that extra oomph. And also, again, there are different concentrations and qualities of lysozyme. I don't know, someone can correct me if you can add too much lysozyme. I mean, I guess at a certain point you will be precipitating out the protein, but go wild in this case. Because whatever lysis, that's you got to get the DNA out. Now, in addition to, of course, the lysozyme is not the only thing doing the job here. This is the first time we're going to use one of the reagents that's provided with the kit, which is this genomic DNA tissue lysis buffer. We're going to add 300, and this is going to make a really important step here to make sure that you are mixing everything well, and pay attention to the way that it asks you to mix things. So in some cases you might shake or flick, but it very specifically says invert. They're likely detergents here, and if you mix it the wrong way, you could end up, in fact, there's a big soap bubble on this when I open it. If you mix it the wrong way, you're just going to end up with a whole bunch of foam, which is not useful. Make sure that you are getting that in there. I should also say, probably there's no reason at this moment now to keep things on ice, because we are about to put this into the water bath. I will try to keep things sterile. This is also a moment, it's not a bad moment to think, if you are deploying this with students, about how would you, or at what occasion do you need to aliquot things. I'm going to already go ahead, I'm going to come back and invert these, but I'm already going to make sure I give them a healthy inversion once I add this. And then the one that I'm most worried about is this really viscous one, because it's probably going to take some time to mix, but I will give it the time to mix. What you're going to be looking for, according to this, is that essentially when your solution is turning clearish, and that could vary from bacteria to bacteria, your lysis is done. So I've added these. These all now have this solution added, and I am going to go ahead and invert, and clearly the one that's viscous doesn't like to be inverted. The other two have these amazing, beautiful golden colors. There's another step, and I guess I'll add it to my teaching temp, and it's, there is a version of this. I was in my mind talking about or thinking about something else that I don't have, because later on there's the opportunity to homogenize these, and I don't have a homogenizer here at my desk, but I do have some needles, and we'll talk about that because we're going to homogenize this later. All right, so I am rigorously inverting this 5 or 10 times, it says. I just want to really make sure that that has a chance to mix. And that viscous sample, I mean, wow, it's quite viscous, but it is starting to mix. And what I'm going to do, it says that we're going to go ahead. So in step 2, for the lysis, I've done A, B, C, which is adding the lysis buffer. And now we are going to incubate it at 37°C for approximately, it says 3 to 5 minutes. So I'm going to do that. I probably won't show you every step of it, but I'm going to do that. But what I'm also going to do, let me find my little floaty there. I'm taking it out. I'm going to go find a floaty. What I'm also going to do is every minute or so, I don't want it to super cool down, but every minute or so, I am going to also give it some agitation, since I don't have a shaking, I'm not using a shaking heated incubator at the moment. Okay, so we'll see you in a few minutes. Okay, so we are back. I was incubating that for about 5 minutes at 37 degrees, occasional agitation. And what you're looking for after that is that the sample has, become a little bit more transparent. These are pretty highly colored samples, but I do notice that they're a little bit more transparent, even our really viscous one. And in fact, actually, they're all a little bit more viscous after that treatment. You can leave it for longer, to an extent. There may be something that never quite completely goes with that treatment, but again, use your powers of observation and optimize it based upon what you are trying to accomplish. So we're now up to step 4. And it suggests that we're going to go ahead and add 20 microliters of proteinase K. So I'm going to go ahead and execute on that. We want 20 microliters. So Really just trying to break things down. At this point also, especially after you have taken things out of the water bath or heat block, this is the time to adjust it. So I'm ramping mine up. The next temperature we're going to need is 56 degrees. And that's a pretty long incubation. I'm going to add it and mix it as well as I can by a number of inversions. That's just my initial set. Once I add it to all of them, I'll invert it a bit more. 20 microliters of proteinase K. Try to start the mixing. Okay, I have added proteinase K to each of these, and we're going to invert it. Now, it mentions that there are 2 more ways to do the next incubation. The next incubation, it mentions if during thermal, we're going to do it at 56 degrees for 30 minutes, which I'm doing because that's the version that doesn't require as much equipment. If you have a homogenizer, you could do it that way. And then after you homogenize, you still end up needing to incubate that for, it says, a minimum of 30 minutes. So again, you probably could decide to go for a little bit longer. Again, we're just trying to make sure that we give an opportunity for these things to really break down. And that's just going to vary based upon what the conditions are. What I'm going to do is I am going to, now that I've inverted, since I'm not going to homogenize, I'm actually going to do another procedure, which is I'm going to shear. And it's an interesting, I guess it really happens at this step, that we're going to take the sample and pass it a couple times through a needle. And that's going to help to break open the bacteria to some extent, for sure. But the other thing that it potentially is going to do is break down the DNA sequence, or rather DNA strands. I'm getting ahead of myself and thinking about the bioinformatics. It's going to break down the DNA strands. Now, one of the things you may know about nanopore sequencing is that one of the advantages of it is the long reads. And a lot of times when you're doing something, especially if you're going to do genome assembly, you want the longest reads possible. And you could certainly get, this is a high molecular weight kit, so clearly you can get high molecular weight DNA from here. But maybe slightly counterintuitively, you actually do want a bit of shearing, because if you do shear the DNA, you are increasing the number of ends of DNA, the ends of the molecules that you've now broken by shearing, and that increases the probability that those molecules will be ligated on, we're going to ligate an adapter, ligate thereafter a barcode. And so it's not to your advantage if you had, millions of, or millions of bases. There's going to be fewer opportunities for ligation to happen, and so lower throughput actually of sequencing. Even though you might get really long reads, it will reduce the throughput. You certainly don't want super short reads, but you can actually get longer. Now, they mentioned that doing the thermal mixing, you could end up with things like 500 kilobase reads, 50 to 500. But if we do the shearing, you can do somewhere around 50 to 250, and I think it's going to help. So we're going to try it. I've got these samples. They've been inverted a number of times. Can't do too much inversion. I am looking and monitoring my water bath, so it's getting up to the right temperature. I've got it on a high temperature to get it up quickly. But for the shearing procedure, I've got my samples. There's no need for them to be on ice, and they're thermally insulated here, so I'm just using this as a more convenient way to keep track of them. And what I have here are some syringes and 26 gauge needles. So these are 26 gauge blunt, I believe, needles. And this is what you want to do for the shearing procedure. Needles are sharp, so just be careful, but it's not the gauge that you normally would see, I hope, in a doctor's office. I guess maybe they use it for something, but I'm not a doctor. And I don't have to be as maybe as thorough as if I'm doing a medical procedure, but I'm going to go ahead and apply the syringe, or get the tip on the syringe. And then usually for this type of procedure, what you're going to be doing is you're going to be passing it through 10 or so times. I think 10 is going to be fine. And it's a matter of just pulling up some sample and pushing it right down. So let's see how well this works. This is so viscous. With this particular sample, I'm getting only a limited amount. For my really viscous sample, I'm going to confess that I'm not really getting anything coming into that. Now, because of that being the case, I'm going to try shearing it with a good old pipette tip. But let's see, I started here with this sample. Very viscous. This is where, if you're familiar with like cooking different gums like xanthan gum and other things. So this is going to be an interesting character. I'm going to try shearing it, although not with a needle. This is live and exciting molecular biology. Let's try with a different sample. Now they are all slightly viscous. Oops, I've got this already. Need another, you're gonna need a sharps container. I'm gonna have to find one. And this is it is a thin needle. This is not a solution that is absolutely thin. Now, I will say though, even if I don't get the shearing to work at this point because of the viscosity, there are other opportunities to do shearing when I'm down to something that's really a more pure DNA sample, because I have all of this slime to worry about. This one is drawing in. It's taking its time. It is going in. You can kind of see how viscous it is. So it's going to take me a few minutes. But I'll go ahead and do it. I've drawn some in, and as you can imagine, getting it out is much easier. One supposes that it will get easier as I go along, because protein molecules are also getting sheared. So actually that worked much better this time around. We're drawing in quite a bit. I don't feel that I need to get all the sample in here, both because of viscosity, and also I don't want to like shoot up with air bubbles. But pressing down goes really well. Now this time it's quicker, so I'm going to go ahead and do that a total of 10 times. I'm looking at my temperature, and I can see it because I'm watching myself on a screen as well. So I've got nothing to do but time for right now. If I have even more time, I'll see if I can go back to the other sample. But in any case, you get what you're doing, so the video will probably get cut here after I've completed the procedure a couple of times. Okay, so much to my delight, I've been able to do needle sharing on all 3 samples. So even number 4, which was quite recalcitrant and viscous, I went in with a pipette and progressively did some shearing with what was obviously a much larger bore opening, and then it was able eventually to get that to work. The other samples you could actually see are really, really clear. There is still the golden color that's associated with probably the carotenoids and that were in the bacteria themselves, but clearly this is on its way to being lysed. So this is going to incubate with the proteinase K for 30 minutes at 56 degrees. Then we have a quick addition of RNase A, and then that's going to be another few minutes, 10 minutes. So this is really the slow part of the protocol, lots of incubations. So I'm going to go ahead and do that, and then we'll see our results after this incubation is complete. Okay, so we have had a 30-minute incubation, and we just have to add the RNase A. So I've got that. After the incubation, really the samples are highly, highly transparent. There is still some of the original coloration, but really looking at it, they look extremely nice and transparent at this point. So I'm pretty hopeful that we've gotten some really good lysis. So I'm going to go ahead and add that. And it calls for 10 microliters of RNase A for each of the samples. And I'll go ahead and do that. One of the things that will happen as it sits for that 30 minutes, a lot of those air bubbles that got introduced in the shearing, those will all sort of settle out. So this time we won't have quite as much disruption, and we'll continue using inversion to make sure things are nicely mixed. Even though these samples are a lot less viscous, there's still some viscosity, and so we just want to make sure that everything gets well mixed. But we don't want to introduce a lot of air bubbles. So let's just gently invert. I'm going to do it 20 times just because of the viscosity, and we really want to make sure it's only going to be a 10-minute incubation. Really want to make sure that RNase A is as well mixed in as I can get it. Okay, I think that's good, and that goes back in at 56 degrees for 10 minutes. While this is incubating, I'm going to go ahead and start setting up the plasticware, and I'll describe that when we come back in 10 minutes. Okay, so at this moment we have completed the incubations, I think just about the longest of the incubations, and we've done the treatment with the lysozyme, we've done the shearing, we've added proteinase, we've added RNase, and again what we have left are really pretty clear, transparent I should say, right, relatively transparent samples. And the next stop is to go ahead and add some protein precipitation solution. So I've got that right here. And we're going to go ahead and add 300 microliters to each tube. And pretty soon after that, it's going to be centrifugation. I can start to get away with reducing my tip waste here and spread things out. This is quite a large volume. And we're going to add 300. So as you can kind of guess, we've got a lot of soluble protein, and we're going to do our best to get that out of solution using this protein precipitation, or separation calls it, solution. And we're going to spend 1 minute inverting. I've got, at least I know where the seconds are on my watch, and hopefully we'll fast forward through this part. Before we fast forward, a note to myself if I'm editing this, what we are seeing as this happens is it goes slightly, especially as it makes, it goes slightly a little bit more opaque. It's hard to see which camera will get that, but it's a little bit white, cloudy, a little bit more opaque. When we do this, the inversion is we don't want to create air bubbles. I haven't done this in a long time, but I do suspect, and reading ahead in the protocol, that we're going to get a phase separation. We're going to get something of maybe a bit of a raft as we try to get those soluble proteins insoluble. So I'm looking at my watch, and it has just about been a minute. And that's going to be followed with a 10-minute centrifuge. So clearly at that point, we'll go ahead and cut. It mentions at that point, really get your plasticware set. And I have taken already a collection tube and then also a bead retaining tube. I guess I'll get a chance to explain a little bit more about how this system works. It's quite clever, I think. And so make sure during that 10 minutes, you just look ahead in the protocol and see what it is that you need to do next. So we're going to go ahead and spin this down. And 10 minutes. Okay, so it has been 10 minutes and we're going to go to the next step. At this point, you could decide if you need the goggles or not, but I think what we have is pretty much splash hazards kind of removed there or minimized. And what we're going to do is I will have a need for some Monarch tubes. So I've already set out these tubes with this little plastic retainer and a collection tube, because there's going to be a number of flow-through steps. And then in this next step, it says that we want a 2 ml tube labeled. So I'm going to go ahead and get those. The plasticware for this kit, as you can imagine, comes with enough for however many samples. And then you can decide again how to appropriately aliquot that for students who might be actually working in teams to do this. Some other things that I need in a moment, I've got a pair of clean, in this case, plastic forceps. And an interesting thing, I guess it's not absolutely required, but I have these wide bore tips. So these are 1,000 microliter tips, but the bore is wider, and that avoids shearing. So at this point, what we're expecting to see is a phase separation with some DNA in an upper phase. And actually now is when they sort of say, worry about the long reads and try not to shear this too much. So I'm going to label these and I'm going to take a look at the separation. It mentions that if there's inadequate separation, you might centrifuge it for longer. So I'm going to see what looks like to my eye, and then think about, do I need to keep this going, or do we see something that looks nicely separated? Okay, I'm seeing a clear, I'm seeing at the bottom, clearly there is protein detritus at the bottom. That was the very viscous sample. In this one also, there's clearly a pellet of material at the bottom. And I suspect this one, it's more along the side. So what it recommends is to be careful of carrying over any of that. You could choose to spin for longer to see if that helps at the highest possible speed. I'm going to go with what I have, and be careful as I can pipetting. It mentions, interestingly, pay attention to it. It says that typically the transfer volume is around 800 microliters, so you may get less than that, and they may be okay if it's a cleaner sample. So let me try it. I'll set it to 800 and see if I get a full 800 or not. With this wide bore tip. And again, I think the viscosity has reduced significantly, but I'm just gonna go careful, be careful and observe, and try not to bring over anything I don't want. It mentions, and just go slowly, it mentions that there is high molecular weight DNA at the interface between these phases. So, for example, I'm seeing like a little wisp that is pulling away as I suck things up. Okay, I've got 800. The sample might have, you know, there's like a little wisp. I don't know if that is detrimental. There's still a pretty clear layer or phase that I feel like, okay, I'm happy with that. That one actually, remember, wasn't suspended. It was suspended in a lot more liquid because we couldn't, we didn't really have a proper pellet in that one. This one, number 19, all seems to be at the bottom. Let's see if that's cleaner. And it also has this little wisp that wants to come up. And I seem to get not a 100%, you know, wisp-free sample, but a clear portion remaining. They mentioned that interface, that's where there's a lot of DNA. But I'm going to take my chances. I think I had a lot of DNA coming in. And also, once we finally get to the end here, there's only so much DNA I can put into the flow cell, and that gets divided because I'm using at least 3 samples here. So I'm just going to do the best I can to get the best DNA. And then this one is more tricky, I'm imagining, because the pellet, it never is not much of a pellet. It's very viscous, and it kind of like rested on the side of the wall. So I'm going as slowly as I can. This one is the dirtiest in terms of there's some wisp material. I am just going to be okay with that. I feel that you can't help it. We are going to do a precipitation with isopropanol, which is going to affect whatever proteins are left in there. And what can I do about that? So I'm going to do my best. I guess the one thing I could say, I think, if I remember any molecular biology, and somebody can correct me, when I add the isopropanol to do the precipitation, proteins are going to be precipitated, but I believe they will be precipitated not quite as quickly as DNA. So in this case, there's going to be a 5-minute incubation or a step of attachment to these beads. And I think, you know, I wouldn't want to let this sit for a long time or go off and have lunch once I've added the isopropanol, because I will probably progressively get more and more proteins coming out of solution versus having a chance at washing them off. Because I've done the transfer, I'm going to go ahead to that next step and just try to go straight through with it. So if it were me, this would be a good pause point if you needed to stop the procedure and maybe put that DNA supernatant at a cold temperature until you're ready to proceed and really try to put the next thing straight all the way through.